Okay, now you can hear me, I guess. I hope you can hear me now. Please let me know if everything is okay now. Perfectly. Okay. Then I'll share my screen, what I'm gonna present, and hopefully now you can also see my presentation. So let's just begin. I'll go to this screen and okay. Yes, you can see my screen. Just shout if you cannot. So today, my name is Janez Konc, and I come from the National Institute of Chemistry in Ljubljana, Slovenia. And today I'm going to talk about our ProBis tools that we are developing within our laboratory for drug development. This is a suite of different tools from web servers, algorithms, databases that we continuously develop for many years now, and they are intended to basically enable you to discover binding sites and ligands on protein structures. So today I thought I will give you first an introduction about our tools. That's gonna take some time, and then we are going to have some hands-on experience, if possible, with the usage of our tools. So why do we need a computer for drug development at all? So this is because we want to speed up drug discovery of new active compounds. So we take a protein structure, and we take a database of ligands, usually small molecules or other proteins, and then we insert these two components in a computer, and we try to find the best combinations of proteins and small molecules which fit together the most perfectly. And the purpose of this computational work is to reduce the number of wet lab experiments which are costly and time-consuming. So we want to minimize this as much as possible using a computer and give just a couple of predictions that are easy and quick to test. So finding a new drug is a difficult task. It's as difficult as finding a needle in a haystack. Of course, this needle is not as big as this one shown here, but it's a very small needle which is at the very bottom of this haystack, and it's very well hidden. So it usually have a lot of work, and we cannot find anything. This is the reality of drug discovery right now. And so we are trying to make it a little bit easier using our computational tools. So I'm going to talk today about virtual screening. Virtual screening tries to starts with a database or a large database of molecules, usually thousands, if not millions of molecules, and we use our computers as a sort of a sieve to sieve through this, to filter out the molecules that are the most promising for drug discovery. So from millions or thousands to millions of compounds, we reduce this to tens of compounds which can be tested quickly within a laboratory and used for and give you useful predictions for possible drug candidates. So the virtual screening can take at least two different forms based on what starting material we start with. And the first one is the first version of this virtual screening is ligand-based virtual screening, where we start with the database of several million compounds, Here we only have small molecules as a starting point. So we have, for example, a known active molecule, a known drug, for example, and we have a database of millions of small molecules, millions of compounds, and we try to find, using this ligand-based virtual screening, molecules within this database that are similar to the active molecule, to the active compound. This is called ligand-based virtual screening. Another form of virtual screening is so-called structure-based virtual screening, where we are better off immediately from the start, because we have the structure of a protein already available. So this is called structure-based virtual screening because we know the structure of the protein that we want to target. This is the so-called target protein then. So we start with this target protein, and we have a database of thousands or millions or even billions of compounds right now, and we try to, using this virtual screening based on structure, we try to fit each compound within each protein structure, which is called molecular docking, to get again a few dozens of the best rated molecules which fit the protein structure the best, and which are the promising for further investigations. So today we are going to go using our tools that we developed through both of these processes, for both types of virtual screening. So for ligand-based virtual screening, as I already said, we need a compound library. Then we need a known active compound, for example, a known drug or a drug candidate or even an investigational drug. And then we need an algorithm that's going to fit our drug onto each of these database compounds. So we've developed a so-called Lisica algorithm, which Lisica stands for ligand similarity using click algorithm, and which is freely available on our incilab.org web page. Here is shown just the download page of our, where you can download the source code and the executables of this Lisica algorithm that we are going to use now in our session here. So I'm going to just go briefly through all these ingredients that we need for ligand-based virtual screening. So first, we start with compound libraries. These are libraries of small molecules. Usually, they can be commercial or non-commercial databases of two-dimensional or three-dimensional structures of small synthetic or natural molecules. So, for example, PubChem is the largest. It has now more than 100 million structures inside, and PubChem represents the knowledge that humanity has gathered about these compounds. They are gathered from different papers, from literature. They are achievable, but they are not usually easily achievable. You have to know the chemistry. So that's why we usually use the other databases that are more commercially available, that offer commercially available compounds. For example, the zinc database is such a database. It holds around 30 million structures nowadays, and you can easily order these compounds and get them synthesized by mail. So you can just say which compounds you want, and you get them, and you can test them in your laboratory, for example. So preparation of small molecules. So all small molecules can be found in these databases as 2D structures or three-dimensional structure, and it depends what you want to do with these structures. Now, if you want to, for example, convert them from 2D to 3D structures, or you have to be careful about conformations, stereoisomerism, ionization states of this structure. So you want to, for example, if you want to have a compound that's active in human or somewhere in human cells, then you have to have a certain pH. You have to take ionization states into account with small molecules that you are going to use. So I'm going to go through our Lisica algorithm, program for ligand-based virtual screening now. So you start with basically a reference compound. This is a biologically active compound, a small molecule, and then we have used, there are different ways to implement this similarity search. So you have a reference compound, and then you have a database of compounds. For example, Zing database, and this Lisica algorithm, which is based on graph theory, enables you to basically find compounds within this Zing database that are similar to your active reference compound. So the hypothesis here is that the similar compounds that you can find in your database, because of this similarity to the known active compound, will also be active themselves. So this is a very simple premise, simple hypothesis, and given that you don't have a structure of a protein, this is enough. But it's usually a hit and miss, a bingo style discovery of drugs that's not really perfect. It's just something that you start with if you don't have anything better available. So similarity between your molecule, your reference molecule, and database molecules is measured using Tanimoto coefficient. Maybe you've heard Jacquard index or Tanimoto coefficient, or it's a very simple measure of the similarity between two molecules. So if Tanimoto coefficient is equal to 1, the small molecules are the same. And if it's closer to 0, then the molecules are very different. So we want to get Tanimoto coefficients which are, for example, about 0.5 or even more or less like this. So you get compounds that are similar to your reference compound, but not very so not completely the same. You understand? So it's a very simple index where you count the number of atoms that are in common between the two molecules and divide them by the total number of atoms minus this common number of atoms. So you get this very simple measure of similarity between two molecules. This is called Tanimoto coefficient. So I'm going to present just briefly how we successfully use this approach to find some new inhibitors that were later shown experimentally to work in a model Alzheimer's disease test. So Alzheimer's disease is a disease of neurons, the degeneration of neurons, in which the acetylcholine concentration is decreased. So in this disease, the enzyme butyrylcholine esterase is increased, and this butyrylcholine esterase makes the disease even worse than it already is, because butyrylcholine esterase degrades acetylcholine even further, so the concentration of acetylcholine decreases even further, and the the disease gets worse. That's why we want to find inhibitors of this enzyme, butyrylcholine esterase, and we use this ligand-based approach to design new inhibitors of this enzyme. So we used active compound, derivative of drug donepezil, as a reference compound, and then we scanned this database of several million of proteins, zinc database, and we found a couple of molecules that are similar to our active compound, and tested them, and they were really active against this butyrylcholine esterase. Here, I want to emphasize one fact, which is not so obvious. I just hinted on it in this discussion about Tanimoto coefficient, which is that we want to get similar compounds to our reference compound, but not too similar. So we want to have compounds that retain some properties of our drug donepezil, but they do not resemble it perfectly. Why is that? Because we want to, in this early stage drug discovery, we want to so-called do scaffold hopping. We want to increase the number of different scaffolds that we are going to test as much as possible in this early stage, because each scaffold gives you sort of a starting point to optimize the compounds in different directions. And you don't know initially which one of these directions is going to be the one that's going to lead to a molecule that can progress to, for example, drug candidate stage. So this is already very, very far. So we want to have molecules that are not too different to our, not too similar to our active compound, but retain some properties of our active compound, which is called in their scaffolds, which is called scaffold hopping. We just hop between different scaffolds. So, and this, our Lisica algorithm enables you to do this. So here is, up till now, we discovered a few compounds that were active, and then we were fortunate enough to have also crystallized these compounds with the protein itself, which was initially not known. And we found that our compounds predicted really mimic the active compound, the donepezil, the derivative, very well inside the binding site. So here we see a binding site inside a protein, and in this binding site, these pink and purple structures are our compounds overlaid over each other as they are crystallized within this protein. So they make up similar interactions with the protein, as you can see, for example, here with this tyrosine and phenylalanine, and they have similar groups at similar places, but their scaffolds are not totally, but considerably different. That's why this is interesting. So Lisica program is basically available as a very Lisica program is available as command line program, which you can use in every Linux or even Windows system. Although Windows system may be not updated very often because we mainly work in Linux, but Lisica software also has this nice user interface that allows you inside a Pinewall molecule visualization program. You can use this Lisica user interface to just graphically define your molecules and your database, and you can see the result of your calculation within a nice three-dimensional molecular viewer. So you input, of course, the reference molecule and the database, which can consist of several thousand or several million of compounds. And as a result, you get here on the left side this list of similar compounds ranked by Tanimoto coefficient. They have these zinc IDs. These are identification numbers of different compounds that are found similar to our initial reference compound. And so here on the left side, you have this list of similar compounds. And on the right side, you can, if you click on one of the compounds in this list, you can see in 3 dimensions how similar are these 2 molecules. The green one is the reference molecule, and the cyan molecule here, the right one here, is a molecule that was found in the database to be similar to our reference molecule. So we designed this interface to be very interactive, very user-friendly, and I will try to later present one example how to use this step by step to you. So now we come to the second part of my talk, which is virtual screening based on the protein structure, or in other words, structure-based virtual screening, which is also called sometimes molecular docking, but the molecular docking is a little bit more, a little subgroup of this virtual screening based on protein structure. We are going to talk about something that resembles molecular docking here. So we are going to start with the structure of the target protein. Here the target protein is known initially, so good, that's good for us. And we have a compound library which can consist of several millions of small molecules. Then it's very important to have a binding site on the protein, target protein, already known, defined. And we developed a ProBis protein binding sites approach, protein binding sites tools that enable you to predict binding sites, the exact location of binding site within a protein if this binding site is not initially known. So we are going to today see virtual screening with the gene ProBis and ProBis for web server. We are going to predict binding sites with these servers and also ligands that can bind to these proteins using our servers. Gene ProBis web server also offers another point, another aspect, which is connected with sequence variants. These are mutations within a sequence of human genes. So sequence variants, we are going to I hope we can still hear each other. Yeah. So we are going to see what sequence variants and binding sites are in just a few slides further. So I'm not going to go into details now. So what are we going to need for this recipe? This is a recipe. These are all like recipes, like cooking recipes. So first we need target protein, which we can get from the protein data bank, which is a database of proteins of crystallographic, NMR, or magnetic resonance determined protein structures, or determined using electron microscopy. And these protein structures in the protein data bank are determined using their PDB IDs. These are 4-letter codes composed of numbers and letters, and each protein usually have several chains also. These chains represent different sequences, usually separate sequences that come together to form one protein. So these chain IDs are usually one-letter codes. Nowadays, a very popular and very groundbreaking thing is this AlphaFold protein structure database, which is a database of modeled protein structures. Now, artificial intelligence, machine learning has come to the point that it can predict us structures of proteins that are within this experimental tolerance of quality. So we have in this AlphaFold database, we have structures for more than 48 organisms, clinically relevant organisms, and we have more than 200 million protein structures. So this in PDB, we have, for example, in PDB, just to give you a feeling, we have 200,000 protein structures, and here we have 200 million structures. So this is a big leap forward. We have structures of whole proteins of different organisms. Protein structures are not static pictures as we get in these databases. They are rather dynamic. They move within our cells, within the solution. They move around, and we can study this process within a computer using the so-called molecular dynamics. So we can simulate how a protein moves, breathes, opens up, closes back again. And now we, instead of having simplified the problem, we have actually made it more difficult for us to select which structure here is the one that we are going to use for drug discovery. One representative structure, maybe the one where this loop closes, or the one where it's open, or maybe we can take all of these frames for drug discovery. We don't know. There is no good answer yet here. So we are here left to experiment for ourselves. So one other very important component that we need to have in order to do structure-based virtual screening is we have to determine binding sites on proteins. So what is a binding site? I'm talking about binding sites, and now I have to define what a binding site is. Here, a protein is shown as this gray ribbon, and another protein is shown as another yellow ribbon, and a binding site is a place where these two proteins come together and they bind onto each other. So they form so-called attractive forces between them that keep them together for as long as it's needed. So a binding site is usually a complementary region on both proteins that enables these proteins to stick together specifically. So this specifically is very important. So it's not just any ligand, it's just one ligand that fits into this binding site usually, or a number of ligands that are similar and they fit into this binding site. So ligands can be other proteins, nucleic acids, or very important for drug discovery, synthetic and natural compounds, ions, and even water molecules. We consider as ligands even water molecules in special cases. So a binding site on a protein structure is usually, if it's for small molecule or for ion, it's usually hidden beneath the protein surface. It's usually within a cavity in a protein. So here it's shown, you can just see a little cavity here on this protein. where there is actually a binding site. And this red actually is the conservation. These binding sites are usually highly conserved regions on protein surfaces. So even though we have discovered many binding sites, pharma industry is using just a fraction of the possibilities right now. So we have to find new binding sites. We have to better define binding sites. In order to drug discovery, we have to have very characterized binding sites, their properties on protein structures. So I've mentioned that binding sites are usually conserved regions on a protein surface. And why is that, actually? If you imagine a protein complex millions of years ago in time, in some bacteria, mutations happen, organisms evolve during millions of years, and those organisms and the mutation can happen anywhere on a protein surface. And if a mutation happens on a region that's within a binding site, it's usually very bad for this organism. These organisms usually die because it's very you have to be really lucky to have a mutation that really makes this protein complex better than it already is. It's usually the protein complex, the ligand doesn't want to bind anymore to the protein, and there is a disease, so the organism cannot live and survive. So anywhere on the protein surface, these mutations can happen, and mutations are just very well tolerated on any part of the protein surface that's not very functionally important. The mutations can happen there and they can be tolerated. But in binding sites, mutations have big effects. That's why binding sites remain similar or the same throughout millions of years of this protein evolution. And our ProBis tools basically exploits this fact to discover new binding sites in contemporary nowadays proteins by comparing a protein. We have an unknown protein, and we have a database of binding sites of proteins. And if we can compare our protein to these other proteins, similarly as we did with the Lisica algorithm before, but on a scale of proteins, and we find similarities between our protein and proteins within the database, these similarities usually mean conservation, and conservation usually mean functionally important regions, and functionally important regions are often within binding sites of proteins. So we can discover basically binding sites by comparing proteins between each other. Here, we don't want to compare proteins globally. We don't want to align backbones, protein backbones, because this is not interesting for us. To discover binding sites, new binding sites, we have to align proteins locally. So we have to find a way to just represent proteins' surfaces as a series of points in space with different color-coded functional groups, and we have to align these vertices onto each other in order to find small local similarities between proteins. Here, in this case, on the right side, we actually found a similarity between proteins with totally different folds, which actually share a similar binding site. So this can be, so local similarity can go far beyond similar folds, similar folding proteins, and can discover similarities, local similarities between proteins, even in proteins that have do not resemble each other on the global scale. And these similarities often indicate similar binding sites. So How do we do actual virtual screening then with ProBis approach? So now I've talked about finding similarities between proteins. So no ligands yet, nothing about ligands. We have just pure proteins without any ligands, and we compare these proteins and we find these similar places, similar regions on their surfaces. But now in PDB, in protein data bank, a lot of proteins that are already in this database already have ligands, co-crystallized ligands on their surfaces. So these ligands can be nucleic acids, small molecules, ions, water molecules, other proteins. So these are all ligands. And now we have our query protein, we compare it to this database, PDB database, we find these local structural similarities here on the right side. These are local structural similarities, and we can then copy or move ligands from the similar binding sites onto our query binding site. So here the hypothesis is that if two binding sites are similar, they also likely bind similar ligands. So we can transfer ligands between protein structures based on the discovered binding site similarities. This is a form of virtual screening that enables us to predict new ligands for proteins that don't have ligands on them yet. So they can be blank proteins, modeled proteins, experimental proteins. They don't need to have ligands, they don't need to have binding sites already known. Because in this similarity search process, we discover binding sites on this query protein and also their possible ligands. We can predict possible ligands of various different types. I'm going to just briefly tell you now how sequence variants come into play now, because we've developed a web server, this gene ProBis web server, that enables you to study how sequence variants, which are mutations of genes in a population, affect ligand binding. So sequence variants, everybody has a genome. We all have genomes, and our genomes differ a little bit between each other. Within a population, there are differences, and so everyone has a set of his or her own unique mutations that define who he is, and all the and also the diseases, and also the things that we have. So they define us who we are. So sequence variants. Otherwise, we would all look the same, and so sequence variants are sort of mutations that happen on genes, and they are more rare, or they can be classified as rare or frequent within a population, or somewhere in between. And they can be something, they can be called synonymous, which are not interesting because they change the they don't change the amino acid residues, synonymous, and they can be non-synonymous if they happen in protein coding regions. And they can be ones that change the amino acids, for example, different meaning, from glutamate to valine, for example, or meaningless. They can just insert a terminal codon inside a gene, so the proteins will abruptly end at one place. So we are interested about these non-synonymous mutations because they have effects on how proteins look like, and we can study proteins with our tools, ProBis tools. So sequence variants are responsible also for various diseases, and they are responsible for our different response to drugs. This is very important because we all respond to drugs a little bit differently, and this is not yet well studied. And using our tools, we hope we can just make it a little more open to the light right now. So, for example, sickle cell anemia is caused by a hemoglobin variant that has one amino acid at position 6, change from glutamic acids to valine. And this small mutation, which changes one amino acid, makes a big difference in how this hemoglobin then associates with other hemoglobin, and how the red blood cells look like, and all the consequences that come with this disease, which are known. So ProBis web server enables you to map sequence variants onto binding sites. So sequence variants come from different databases. You can find it is DDSNP from the NIH, Cosmic Ensemble. Different databases have all these sequence variants, which can be somatic or so population within population. So not important. So we start with sequence variant. We go from there to gene. This mapping is already known, and then we do a mapping from genes to structures using ProBis web server, and we map the sequence variants onto the structure of a protein. So when we have a sequence variant on the structure of a protein, we can say, okay, this sequence variant looks dangerous because it's close to a known binding site, ligand binding site. And this ligand binding site binds, for example, an endogenous substrate of this protein. That's a problem. So we are going to have a problem. This sequence variant is going to likely affect our ligand binding. And this is the added value that our tool gives you. Because using only gene, we cannot tell if this is a ligand binding site or not. But if we map a sequence variant onto the structure of a protein, we can easily see where with what ligands this sequence variant can interact. So in our web server I'm going to show you briefly after this talk, we can just see variants in a, for example, protein sequence, which are the sequence is shown here of one protein, and these sequence variants are red amino acids in this sequence. And you can also see the mapping of different binding sites onto the sequence of a protein. So we have we can just, for example, here we can see this arginine R amino acid sequence variant arginine is actually in two binding sites, and this is a nucleic acid binding site and a protein binding site. So this one might be very dangerous actually. Arginine to serine, it's a big change, and also this sequence variance is in two different binding sites. So it might affect binding of two different ligands. So this might be problematic sequence variant. So in ProBis web server, these sequence variants are red, usually red residues, and you can just see here maybe the structure of one ligand that's shown here and how the sequence variant interacts with this one ligand, predicted ligand of this protein. So nowadays, AlphaFold is very important. Also, Facebook has developed an artificial intelligence method that can predict millions of proteins of different organisms, but we are going to concentrate now on AlphaFold protein structures. So These protein structures are very big achievement, but they are blank protein structures. They don't have ligands. So they are just proteins without anything. There is nothing more about where the ligands bind, how the ligands bind, which ligands bind to these proteins, and we are going to trying to address this question. We are trying to solve this by using our ProBis fault web server that I'm going to also show later. So we take AlphaFold protein structure model as input. We compare it against the PDB using ProBis algorithm. We find these local structural similarities between AlphaFold protein structure and PDB structures. And then we can transpose or move the ligands, as I already explained, from the PDB to the AlphaFold protein structures. So these blank AlphaFold protein structures can then get their ligands finally, and there we can determine different ligand types, such as proteins, peptides, nucleic acids, small molecules, which can be further divided into cofactors and compound, small molecules, metal ions, waters, and glycans. Of course, glycans are, of course, covalently attached to a protein, but we can still predict their locations using our similarity-based approach. So, we can basically get all these different ligand types onto a surface of a protein. And how do we then define a binding site? We then cluster these ligands of the same type by proximity, and ligands that basically overlap between each other represent one binding site. So, I'm not going to go into detail. We then refine small molecule binding sites into the substrate competitive binding sites, cofactor competitive binding sites. We calculate different scores, druggability scores for substrate and cofactor competitive binding sites. Druggability scores tells you how amenable, how good a binding site is for drug discovery. So a binding site with a high druggability score, we are going to try to use for drug discovery, and the one with low druggability score, we are going to maybe use it later for drug discovery when we don't have better options yet. So the result of this comparison and ligand transposition and clustering and scoring are predicted binding sites and predicted ligands for each binding site in AlphaFold protein structures, and we took human proteome as the start. So human is, of course, the most important organism according to our standards, and we wanted to have a human proteome mapped with all these binding sites and ligands. So this is just to show you that we can find new binding sites useful for drug discovery that are not in the protein database yet. In fact, there are many proteins in AlphaFold protein structures that are not very similar to PDB structures, and we can find druggable binding sites even on those proteins, new binding sites, which are highly druggable, likely druggable, and so on. So I'm going to just briefly go how these protein binding sites look like. So this is, you've maybe seen this blob, this surface. This represents one binding site. So a binding site in our web servers, binding sites are basically not just amino acid residues, but the volume taken by all these possible ligands that bind to these binding sites. So ligands basically define where a binding site is. So on a surface of a protein. So this surface represents one binding site. So this is a small molecule binding site, for example, this blob here. And all those possible predicted ligands, below these binding sites are all these possible predicted ligands, which we can just turn on and off, and we can see them how they look like in 3 dimensions. And protein binding sites, we can also predict protein binding sites. And we can also predict binding sites for water molecules and ions. Water molecules especially are very interesting because some of the water molecules actually have important structural and functional roles within a protein. So they do not just randomly bind to a protein, they always bind to the same location on a protein structure. So a water molecule just goes into the protein and just stays there, basically. And using its very strong forces, it sticks the protein together and holds the protein structure together. And sometimes it has also a big effect on ligand binding, because ligands can make interactions through water molecules with the protein, not directly to the protein, but through water molecules, through one or several water molecules. So, and we can predict, using our tools, we can predict such important water molecules within our process. We are going to see the use of ProBis for web server on a small example later. And you can just, for example, you can download the binding sites for entire human proteome. For example, small molecule, compound binding sites, cofactor binding sites, peptide binding sites, you can just download them for all the human proteins that were modeled using AlphaFold method. We have predicted for more than 20,000 protein structures, all these binding sites that you can download and use them within your projects. For example, one possibility is to use them in inverse molecular docking, molecular screening, is when you want to, for example, take a natural compound and screen it against the entire human protein. So you can take our database of binding sites and you dock, for example, your small molecule within each of these binding sites and see and predict where your natural compound will bind to which proteins, what are the target proteins of your natural compound or some other compounds that you are interested in. So, since you don't have, I didn't give you this installation instructions yet, I'm going to do these exercises myself. You can try to exercise one. You cannot do because you don't have PyMol probably installed and you don't have Lisica plugin installed, but it's very easy to do, and I'm going to show you how to do it. And in exercise 2, I'm going to use the ProBis web server to do virtual-based screening based on the protein structure and find some sequence variants within binding sites. So in this first exercise, our objective will be to predict drug candidates based on known ligands, based on known drugs. We are going to use our Lisica algorithm, which is available at this incilab.org/Lisica address under the Downloads tab. And we're going to choose one of the drugs here listed here and download its structure in Mol2 format. And we're going to run the PyMol program with Lisica plugin already installed and do a small comparison of our molecule against the example database of about 14,000 molecules, which is going to be very quick, as you will see. And we are going to try 2-dimensional and 3-dimensional virtual screening using our Lisica software. I hope this will work. I hope this will work. Now, because I have really tried that. And I'm going to have to share another screen with you right now. Let me see. I'll share another screen. Desktop. Here okay, here you're going to see the my terminal, probably. I hope you can see my terminal now. And I'm going to just let me see. I have already a couple of drugs in this terminal, and I'm going to start a panel program, which I don't know if you will be able to see. Otherwise, you cannot. So I'll just make another different sharing. I'll make it happen another way. Entire screen. Okay, this will be probably the I'll just share the entire screen with you, and whatever will be on this screen will just be able to see, hopefully. So you can see Python program, which you probably already know, because it's very well known. You can get it for free from this Schrödinger software if you are a student. And here I just went to this plugin, which I already installed. I already installed our plugin, which is available here. Let me just show you where you can get our plugin. It's lab.org. And you can go to this seats. We have all these tools available. This is a plugin, and you can download it here. It's very well updated, so it really should work with the latest primal version. Basically, when you install it, this plugin will appear here in the primal under the plugins menu, and you can then have, for example, open molecule, your reference ligand. We were talking before about reference ligand, so we'll take Paxlovid antiviral compound and a database of molecules, 14,000 molecules that I downloaded from INSILab.org web page before, and we're gonna go for two-dimensional virtual screening and just press go. Instantly, 14,000 compounds is no match for our algorithm. It's used to millions, if not billions, of small molecules. We got a similarity. So we got we got compounds ranked from the highly similar to the less highly similar compounds that you can just see side by side. The green one is the reference compound, and the cyan one is the database compound. So you can just get 10 motor coefficients. Here is not very high, 0.4. So these compounds are not very similar, but this is just because our database is very small, basically. It's just a random database that's not optimized or not big enough to get compounds that are really similar to our Paxlovid drug. So you can just see which atoms, for example, are similar between our compounds, and you can then, for example, run another three-dimensional virtual screening now. This example will look a little different. Now we'll have our reference compound and database compounds overlapped. So this is what our algorithm found. That this is the best similarity between these two compounds, which is not basically very good. But for sure we achieved here scaffold hopping, scaffold hopping, so we retain some similar properties of our reference compound, but we have a different skeleton of our, we've discovered compounds that have different skeletons than our reference compound, which is what we want to achieve in ligand-based virtual screening. So this is Lisica software, and I'm going to go now. Let me see. I hope I'm still sharing. Yep, am I? I'm sharing, and then I'm going to go to our ProBis web server. ProBis web server, which is here, available here. And we are going to do something else here. We are going to go to exercise number 2. I'm just going to show it like this. Exercise number 2. Here we are going to try virtual screening based on the structure of the protein, and our objective will be to predict drug candidates based on the structure of the target protein in ProBis. So we are going to have this ACE2 protein. You probably know it. It's a protein that's involved in COVID. It's a human protein that basically binds to the COVID protein, the spike protein, and we are going to find predicted ligands that bind to this protein, and we are going to find one drug among these predicted ligands, and we are going to try to find the binding sites for SARS-CoV spike protein on this ACE2 protein. And also determine which sequence variants on the ACE2 protein can affect SARS-CoV binding protein spike. So I hope I will be still able to do this. It's ACE2. Here we are just typing in the gene code, the gene name of this protein that we are interested in. Sorry, we have to type it again, ACE2 protein, submit, and we are going to get. So this is our server gave for this gene. Basically, we have a many possible protein structures. So we'll just go with the first one. They are probably very similar. Maybe with some details different, but we are going to just use one ACE2 protein for this example here. So what it did now, we predicted the binding site of this protein. This block here, the green block, is a binding site on this protein. And this red amino acid residues are sequence variants on these proteins. Red ones are the ones that are near the binding site and can affect binding of ligands to this binding site. So let us see what else we can find. Table of binding sites here. Let's see here. So we have all these different possible bindings. This is green one is the compound binding site. So these are glycan binding sites, ion binding sites here inside the protein, protein binding site. So we can predict all these different possible binding sites on this protein. But for example, our goal was now we have a binding site. So now we go to a different table of ligands. So here I predict the ligands for these binding sites. Yeah, and if we click here to the add select, we can just turn on the ligand. You can see, as you can see here, the ligand here is now shown here in this binding site as a sticks model. So very easily we can just see which ligand binds to which binding site, and we can go to this binding site number 2, and we can go through all these different ligands, and I know already which ligands are these are ligand codes, and we're gonna just turn on some ligand that's not that resembles one drug, for example. So this is a ligand. I think it's a derivative of some antihypertension drug, antihypertensive, and we can see that this ligand interacts with one sequence variant. So we can predict that sequence variant will be likely important for binding, correct binding of this ligand. If we have some other protein, so this sequence variant means that here we have a histidine, but in certain percentage of our population, this will be arginine, histidine arginine. So this change is, of course, quite a big change in interaction. So accordingly, people with arginine may have better or worse binding of this ligand of this drug to their protein. So it may affect the more or less than general population. So you see, this is very important. Sequence variants are very important, and using our tool, we can just determine where they are on the surface of a protein and what ligands they interact with. So we wanted to also find which protein which are protein ligands, what are protein ligands of our ACE2 protein. So we can turn on one protein ligand. Let me see. Let us just turn on this one. So, and this is actually a spike protein. So here the spike SARS protein binds to our ACE2, and this is how this interaction that's called the SARS. This is SARS, not SARS2. Actually, how this virus attaches to our protein and acts on the protein. So we can see that there is one sequence variant within this glycine to valine, which might affect binding of SARS onto this protein. This might, for example, knowing sequence variants in this type of binding site may be used to predict that some people are more prone to developing the disease, or some are more immune to the disease, and not just the SARS, but also, of course, other possible diseases that we know are caused by protein binding. So the gene ProBis enables you this, and then I said I'm going to also show you, for example, the ProBis 4 web server let me see. ProBis 4 web server enables you to so has all these human AlphaFold protein structures within them, and all these predicted binding sites are in this database for these different ligand types. So you can just, for example, here are just examples, conserved waters. Let us focus on conserved waters right now. So these are all these proteins, and we have water sites. One water site on this protein, one water site on this protein, 47 water sites on this phosphatase protein. Let us see this protein. This is interesting. Let me turn on this binding sites. We just look binding site viewer in 3D. So these AlphaFold proteins are composed of regions that are very well folded, and some regions around them that are not very well folded. These are these ribbons around the protein. They are not accurately determined. And here you can see a water site within this AlphaFold protein, and you can just turn on the water molecule. Here is the water. This red thing is the oxygen of water molecule, and this is the predicted place that will bind to a water molecule. Let us see also a compound binding site on this protein. This will be here. These are not really drug-like. Drug-like binding site. Druggability here is unlikely druggable. Here the binding site is unlikely druggable. So we'll but we have a binding site for a small molecule that we predicted with the corresponding ligand within this binding site. A protein binding site. Let us predict a protein binding site. Here, protein binding site and a possible ligand of this. This gray ribbon is the predicted ligand of this protein. So this is what ProBis 4 can do for all the protein structures in the ProBis database, AlphaFold database. And we have also, I must not forget, we also have our basic ProBis web server, which is very heavily used. And I don't know, I hope this one basically gives you the similarity between two proteins or a protein and your protein database. So we'll just take an example protein, and let us see. Okay, we have pre-calculated results for this protein already, so we'll just take them from the database. Otherwise, you will just click submit job and wait for about a couple of minutes to an hour, depending on the traffic. But let us see. Okay, something not working here very well. No, it is. Is it just slow, or something did not go very well here? So we should just try the live computation, but this might take too long. Okay, so it's working. So it will work for quite a long time actually. So we won't see, but you can get here in this ProBis web server. You can get you can input your query protein, and you can get similar proteins, locally similar proteins found in the protein database. So with this, I would like to thank you for your attention. And if you have any questions, or maybe have a time, a minute or two, for some questions. We have already some questions here, which I found. Do we get an estimated volume for the protein binding site get generated? Volume as a number, no. But we could easily calculate this volume because we can download all these binding sites. So the question was, do we get an estimated volume for the protein binding sites generated? The answer is no. It's not on the web server, but you can download all these binding sites, and you can have these spheres, a set of spheres define a binding site. They define a binding site, and you can calculate, not so easily, but you can calculate the volume, approximate volume of your binding site. But even approximate volume of your binding site by counting the number of spheres within each binding site. It's very easy. You can just count how many spheres a binding site has, and you can see, okay, it's small binding site, so it's big binding, not very precisely, but still you can estimate the volume of a binding site. Different web servers give different binding site residues for the same proteins. How do we identify the correct one? Different web servers give different binding site residues for the same protein. How do we identify the correct one? Yes. It's not using our ProBis web server or some tool, ProBis, Fold, ProBis dog database, gene ProBis, gives you one answer. Usually, our tools give you the similar solutions between our web servers. But of course, you can have a different tool that gives you another. Sorry, we lost connection for a minute because somebody called me on a phone. So it seems that it disrupts the connection here, and so it's not. So basically, other tools gives you another solution, and basically you have to see different tools and get a consensus. If there is a consensus, you can just between different tools, you can just probably believe it more that there is a binding site there. And otherwise, our tools are precise in the sense that what you get is evolutionary based binding sites based on evolutionary conservation. So it's not something that's machine learning or something that can get you. I mean, it's it should be very precise actually. Yeah. So based on this evolutionary conservation, it's not. It should be the binding site should be there actually. There is also one question about off-target prediction. So basically, you can use our tools binding site. The binding sites from ProBis 4 web server or ProBis doc database. I didn't show that database today, but there is another database for the PDB that gives you all the binding sites within a PDB, and you can just download all these binding sites and use your favorite molecular docking tool to dock your compound against all these possible binding sites and get a ranked list of binding sites, which is possibly some of these binding sites on the top of the list ranked high are off targets of your drug. If they're not, if this is not a target protein, something that our tool can give you a prediction of off targets this way. Another way that we can predict off targets is that if we know a binding site, a true binding site of our small molecule drug, and this binding site is within a PDB, we can just compare this binding site using ProBis web server to all the PDB structures within the PDB, and we find similar binding sites to our known drug binding site, and this similar binding site might be off targets for our drug as well. So there are actually two different approaches that we can use to find binding sites here. This one with ProBis web server and similarity detection, or binding site similarity detection, is very simple to use. You just input your protein into ProBis and you get similar proteins, and the ones on top of the list are probably off targets for your drug that you are investigating, that you know binds to your query protein. Another way is a little bit more complicated. You can take our database of binding sites, either for the AlphaFold database or for the PDB database, and you can use another docking program to just scan your virtual screen, your compound against all these different binding sites. Okay, I hope this answers your questions, and I would really like to thank you for your attention, that you remain such a long with me for this talk. I have to get going right now, and I hope to. If you have any more questions or about our tools or anything else, you can just give me. You can just mail me on my cons. And I'll also Fidel will give the slides to Fidel, so you can see my slides later. Okay, thank you very much. And I have to be going. Bye bye. See you.